We are coming to you live from Purdue University with our It's a Gene Thing zipTrip. I'm Jessica, your Purdue zipTrip guide. Now we have students all over the country watching this broadcast. Just take a look at this. Hales Corners Lutheran Middle School. Are you guys there? Go! Yeah, I love to see that pride. Are you guys ready to have some fun today? Yeah. Cool. Okay, we'll be checking in with you periodically throughout the show. And right here in the studio, we have students right here in Indiana joining us from Southmont Junior High School. Hey, guys. Hey. Don't be shy, don't be shy. Well, while we cannot see all of you out there, today's zipTrip is interactive, and you have a chance for your voices to be heard. Now, if you have a question for any of the scientists, you can email them into us here at the show. We're using a new way to send email. It's through our new website. So for all of you teachers out there, on your My zipTrips page in the It's a Gene Thing show, you will see the green box on the left side of the screen where you can submit your questions for the show. Just type in the question and hit Ask. You can ask as many questions as you want, and don't worry, if we cannot get to your question today, a few days after the show is over, there will be a link to all of the questions asked with answers from our scientists. And we are also using another great interactive technology today called Purdue Hot Seat. And let me tell you, it is hot, hot, hot. We're using it to take student questions, so everyone say hello to our Hot Seat correspondent, Abby. Hey, Abby.
Hello, everyone. Hot Seat's a social media program that lets us use computers, cell phones, and other mobile devices to post questions and vote on the ones that you students would like to see answered on the show. The Hot Seat is packed today. Joining me here, we have River Valley Middle School, St. Francis and Clare, Hales Corners Lutheran Middle School, St. Thomas Aquinas Regional School, Andrew J. Brown Academy, Noblesville West Middle School, McDuffie School, Hendersonville Middle School, and St. Thomas More School. Now, Hot Seat users, be sure to answer the poll question before moving on to topic number 1.
Wow, we've got a lot of schools joining us today, and thank you, Abby. We look forward to hearing from those schools throughout today's program. Now, this zipTrip is all about genetics. Do you know what a scientist who studies genetics does? Well, today we're going to find out. First, we are going to meet a young geneticist who's on a team researching the mechanism that allows some plants to thrive in soil with high levels of poison like arsenic. Then, we'll connect with a laboratory right here on campus where something fishy is going on. Finally, we'll work with Purdue wildlife experts, outdoors and indoors, to use our scientific inquiry skills to help solve a genetics case about these unique amphibians. So, get ready to meet some scientists, have a little fun, and learn something too. This zipTrip is all about genetics and the mystery of life.
Come take a look at this crick.
What is it, Watson?
I think we're onto something. What does this image of deoxyribonucleic acid, or DNA, look like to you?
Well, I believe it looks like a long, twisted ladder.
Yes, yes. It appears that DNA looks like a double helix. See the two phosphate backbones? And the base pairs are like the rungs of a ladder.
Let's build a DNA model with simple materials from the lab.
Totally.
And show the world that DNA, the very building blocks of life, are like a twisted ladder.
This is so exciting.
Yeah!
This is so so exciting. Yeah! Well, as we're learning today, DNA is the genetic material that determines a plant or animal's characteristics, like the color of leaves, or for humans, the color of a person's hair. And scientists who study DNA and all of the different structures and environment that surround it are called geneticists. There are many geneticists working right here and studying here at Purdue, and joining me now is one of them, Nadia Atala. Hi, Nadia.
Hi, Jessica.
Thanks for being here today.
Thank you so much for having me.
This is your first time with zipTrip, so we're so lucky to have you. Now, you don't look like a normal scientist, so what's your story?
Well, I'm a scientist and also a graduate student studying plant genetics here at Purdue in the Department of Botany and Plant Pathology.
Very cool stuff. So are studying the genes of plants much different than studying the genes of humans or animals?
Well, the fundamentals of genetic research are the same, going back to basic Mendelian genetics. For instance, we all start out learning the Punnett square. Do you all know about the Punnett square?
Do you guys know anything about the Punnett square? Looks like we may need a refresher.
Right. So here's a quick example on how a Punnett square works. This one's using mice.
Great, let's take a look.
The Punnett square is a diagram that is used to predict an outcome of a particular cross or breeding experiment between two individuals. The Punnett square is a visual representation of Mendelian inheritance. Genes are either dominant or recessive, and we will represent that by an uppercase letter and a lowercase letter. In this Punnett square, we'll use the letter B. We will use mice as an example in this Punnett square, and the big B will mean dark brown fur, and the little b will mean white fur. There are 4 sections to this Punnett square, with each section representing an outcome of 25%. We have 2 light brown mice. We'll be determining the probability that the offspring of these 2 mice will have either dark brown fur, light brown fur, or white fur. Both mice have 1 big B, which gives dark fur, and 1 little b, which gives white fur. Mating these 2 mice gives us 1 square with 2 big B's. This gives us a brown mouse. We get 1 big B, little b, which gives us a light brown mouse. Then we get a little b, big B, which also gives us a light brown mouse. And finally, we have a little b, which gives us a white mouse. And that's how a Punnett square works.
Well, that was a great quick genetics lesson, but you are a plant geneticist, so can you tell us a little bit about what you're studying?
Sure. So I have 2 areas of research. The first is it focuses on plant development.
Okay.
So specifically sex determination in plants.
Okay.
So how the sex of a flower or plant is determined is very important. In humans, we know that sex is determined through sex chromosomes. However, plants have a wide variety of mechanisms, from sex chromosomes to sex being determined by the environment.
Wow, the environment.
Yeah. So the plant we study we focus on a fern, Ceratopterus.
Okay.
So ferns have been around for a very long time, since even before the dinosaurs.
So they're like prehistoric.
Yeah, exactly.
Cool.
And so because of this, and because seed plants, such as the crops we rely on today, evolved so much later, we can learn a lot about seed plant development by studying more basal groups of plants, such as the ferns.
Okay.
So in our lab, we use a variety of techniques to study Ceratopterus. I use a mix of molecular biology, computational biology, and reverse genetics.
Which is what we're seeing right now.
Yeah, so forward genetics is when you have a phenotype and you want to find the gene that's responsible for that phenotype.
Okay.
Reverse genetics, however, is when you have the sequence of the gene and then you want to see what that gene does. So we use reverse genetics in our lab in that we have the sequences of the genes and we want to find which of these genes determines sex in Ceratopterus. So we do a lot of knockout experiments, and so we knock these genes down and then we look at the phenotype. Knockdown experiments can tell you a lot about a gene's function. For example, if I'm studying a plant that makes purple flowers, and I want to know if gene A is responsible for creating the purple pigment, I can knock down the expression of gene A, and then if the plant starts producing white flowers, I know that there's a really good chance that gene A was involved in some way in making the purple pigment.
So you solve that purple pigment mystery.
Exactly.
Awesome. That's really interesting work that you do. So how will what you learn be applied to your future work?
So overall, we want to uncover how sex determination occurs in Ceratopterus. However, then we can apply this research and knowledge to seed plants. So how sex is determined in plants is very important and has profound implications in agriculture and plant breeding, because the sex of a plant determines what other plants it can be bred with, and in some cases even cultivation.
Okay.
So you see, genetics can unlock a lot of processes for us, can improve agriculture, can improve plant breeding, and can even lead to the creation of plants with new traits, such as drought tolerance or the ability to tolerate toxins in the environment. And that brings me to my next area of research.
Okay, and I see you brought some plants with you today, some little friends. So what's their story?
So I'm working on a team of researchers here at Purdue that is studying this fern, Pteris vittata. So and what's neat about this fern is it has genes that allow it to hyperaccumulate and tolerate large quantities of arsenic in the soil.
Arsenic, like the poison?
Yes, so arsenic is a highly toxic element, and arsenic contamination is prevalent in many areas of the world in the soil and water. It can be naturally occurring or it can be a result of human activities. And this arsenic contamination often leads to arsenic poisoning.
You're right.
And so we're studying this plant because it can accumulate and tolerate arsenic. It tolerates 100 to 1,000 times more arsenic than other plants. So the amount of arsenic that this will thrive in will kill plants such as this Venus flytrap and quickly kill them.
Wow.
And so we are studying the genes that are involved in this response, and because this plant also can literally suck arsenic out of the soil and store it in the fronds here.
That's so cool.
So we can hopefully use these genes and put them in other plants to allow these plants to suck arsenic out of the environment, and then we could maybe use these plants to remediate contaminated soils.
So the fern is basically like a toxic savior, like a superhero almost.
Yes, it's a superhero fern.
That's really amazing. Well, thanks for telling us all about your work today. Before we let you go, I hear we've got some hot seat questions for you. So Abby, what's the hot seat question of the moment?
The hot seat question of the moment is, why does DNA look like a twisted ladder?
So DNA is a double helix. It has a sugar phosphate backbone, and it has complementary base pairing. So it's all about the structure of the DNA, and that's why it looks like this double helix. So the base pairs, you have A pairs with T, G pairs with C, and then you have these on this sugar phosphate backbone. And it has minor grooves and major grooves, and that's why it has this twisted shape.
Very cool stuff. We just also got an email question that came in for you from Blake. He is at West Middle School in Nobles, Indiana, and the question is, if the sex of a plant is determined by environment, will changing the environment change the sex?
Yes, absolutely. So in the plant that I'm studying, Ceratopteris, the female plants release a pheromone that turns all the developing plants male.
Really?
Yeah, and so then if you take one of these male plants and plant it somewhere by itself away from these female plants, it will revert back to a female.
So will that happen with male plants? Will they be able to turn a female plant male?
No, not in this
versa?
species, no. Yeah, it's only the females have the ability to do that.
Wow. Go females. All right, so let's check in with Hales Corners School. Do you guys have any questions for Nadia?
Yes, we do. In your video, you mentioned reverse genetics. So what exactly is that?
So we start with the sequence of a gene, and we have the sequence, but we don't know what the gene actually does. And so what we do is we try to figure out what the function of this gene is. And one way you can do this is by lowering the expression of this gene or knocking it down, and then looking at the phenotype that we see. So it's kind of the opposite of what you normally think of when you think of genetic experiments, which is known as forward genetics, when you see a phenotype and you try to find what gene causes that phenotype.
So total reverse. Thanks for your question. And let's go ahead and check in with our studio audience. Do you guys have any questions for Nadia? Yes, you, right in the center.
About how long does it take before you can determine the sex of a plant?
Good question. About how long does it take before you can determine the sex of a plant?
That's a very good question. It varies by species. So in the fern I'm studying, about 6 days. However, in some plants, such as date palms, it can take over a decade. And that's why understanding the mechanism of sex determination is so important, because it's really hard to breed those plants that you don't even know what sex they are for a decade.
Awesome. Well, thanks so much, Nadia. We're going to check back in with you at the end of the show, so stick around. And if any of you think of more questions, just submit them through our website. Wait, so you mean to tell me that we are all mutants? I know I looked pretty spooky in that video. Now, before we go to our next scientist, I have a quick genetics activity for us to try. So do this with me. I want you guys to pull on your earlobes like this. And I want you to see if your earlobe is attached or detached. What about you? Is it attached?
Attached.
Attached? Mine's detached. And now I want you guys to stick out your tongues. I know this is silly. And I want you to see if you can roll your tongues like this. Anybody? Some of you? Some can't, some can. Okay. Well, let's go ahead and check in with our hot seat and see what they are saying. Abby.
Our poll shows that 75% of our hot seat users have non-attached earlobes, while 25% have attached. And 75% of our hot seat users can roll their tongue, while 25% cannot.
Okay, so I guess I'm in the majority with the tongue rolling thing. Now, we've learned that thanks to genetics, we have physical traits like different eye color or hair color. But have you ever thought about why we all have different personalities or are more interested in some subjects than others? For example, why do some of us like to travel, like this girl right here, and some of us don't? Let's take a look at this.
How's it going?
Great, I'm so glad it's the weekend.
Me too. Do you have any big plans?
No, not really. I'm just gonna, stay at home, play some soccer, maybe hang out with my brother, watch TV, get on Facebook maybe.
Well, if you get bored, you should call me. A bunch of us have been planning to go to the movies and maybe get ice cream afterwards.
It's okay, I really need to get caught up on some TV shows.
Tomorrow, Sarah and I are going to the festival downtown and then maybe go shopping. You should come with us. It'd be really fun.
I really just like to stay home and hang out on the weekends. I mean, don't you like to take it easy and relax?
No way. I like to get out of the house, hang out with my friends, and do different things. So if you get bored, just give me a call.
Oh, okay. Well, there's my mom.
See ya!
Well, just like humans, there are some animals who like to travel and some who don't. A scientist here at Purdue is trying to find out more about fish and what makes them swim in some pretty unique places. Joining us live from his lab in the Lilly Hall of Life Sciences here on campus is fish geneticist Matt Hale. Hey, Matt.
Hey, Jessica.
And can you wave hello to everyone who's watching?
Sure. Hey, everyone.
Hey.
Hi.
So, Matt, can you tell us a little bit about where you are?
Sure. So I'm in the Lilly building here at Purdue University, the main campus in West Lafayette. And here in the Lilly building is where the Department of Biology is kept. And here there are many different scientists working on answering many different questions in biology, from some people who work on cancer, some people work on photosynthesis, and some people work in animals. So we've got people here working on amphibians, some people work on birds, as well as several different scientists who work on fish, like myself.
That's really cool. Your lab sounds really cool and very high tech, but you're not always in the lab, right?
That's right. So we have a facility about 5 miles to the west of campus, the aquaculture facility, which you guys are seeing some footage of right now.
Yeah.
So here there are several different researchers who keep several different species of fish alive, and that lets us ask and answer several different questions about the fish's biology or about their behavior. So some of the footage you're seeing now is of one of the 2 species of fish I work on. These guys are called brook trout. And brook trout are found throughout the Great Lakes region of Canada and North America. And some of the questions we're looking at with these guys is why is it that some of them seem to stay at home, whereas others of them migrate. And some of these guys migrate around Lake Superior and Lake Huron before returning to breed. And so what you're seeing now is some of the footage of some of the fish. We weigh them, we measure them, we can cross them, so we can take the eggs from one fish and the sperm from another fish and cross them to make a new generation of fishes. And we can keep looking at these fish and measuring things like their size or their weight and seeing how that changes from generation to generation.
Very cool stuff. So we're learning that scientists use different animal models to learn more about different types of genetics. So Matt, can you tell us, of all the animals in the whole world, why did you decide to work with fish?
So when I was about 8, I got a fish tank for a birthday present from my parents. And that kind of really started my interest in fish. And I had things like guppies and swordtails, the sort of things that you can see nowadays in pet stores.
Sure.
But I've been interested in migration for a long period of time. And many different species of animals migrate, from birds to mammals and insects. But often it's really difficult to know whether or not an individual has migrated or if it hasn't. And with the fish, with the salmon and the trout, it's really, really easy to tell if an individual has migrated or not because of their appearance. The size of the fishes are very, very different.
Gotcha. So they're great creatures to study. Now, your job takes you to all sorts of cool places, and I hear you have a really cool project going on right now. Can you tell us about that?
Yeah, definitely. So the main project I'm involved with here at Purdue is out in Alaska, not far from the town of Sitka, which you guys are seeing some pictures of now. So this is really, really remote. You can't get there via a car. There are no roads. You've got to get there by a float plane, these small float planes.
Wow.
And what we're doing here is we're looking at a different species of trout. We're looking at a species called rainbow trout. But just like the brook trout, they have two different types. There are types that stay at home and types that migrate. These big guys, these big silvery guys, they migrate out into the North Pacific for 3 or 5 years before they come back to spawn. Whereas these smaller, colorful guys with the pink band along the center, those are the residents. They stay in the streams that they're born throughout their whole life cycle. And one of the cool things about this system is that we can catch the fish as they're coming back. So now you're seeing some of the drift nets we put out, and when we put these out right as the fish are coming back from the open ocean to spawn. And that lets us catch all the migrant fish as they're trying to get back into the streams to spawn. What we can also do, as you're seeing now, is we can take boats upriver and catch some of the resident fish, the ones that don't migrate out into the ocean, by just basically using a fly and reel. So just going fly fishing and collecting the fish.
So you basically have the homebody fish, and then you have the thrill seeker fish.
Yep, you got it.
So how long were you in Alaska?
I was in Alaska for about 2 weeks, right at the end of May and early June.
Awesome. And then how big was your team?
There were about 5 or 6 scientists who were up there pretty much throughout the whole breeding season. So when the first fish start to come back in the middle of May, through to about June the 20th or there and thereabouts.
So do you bring back information from the fish in Alaska to your lab here at Purdue?
Yeah, definitely. So as you saw some of the footage there, we take things like the weight and the length of the fish, but we also take a tiny piece of tissue. We take a little fin clip from each of the fish we sample.
So a fin clip. So just to be sure, that doesn't hurt the fish, right?
No, not at all. It's just like taking a fingernail clipping. It doesn't hurt the fish one bit.
Awesome. And so what do you do with the DNA from the fin clips?
So once we get the fin clips back here to Purdue, the first thing we do is we extract the DNA. And then once we've done that, we set up a whole bunch of reactions. I'm going to show you an example of right here.
Okay.
So just make sure you guys can see this.
Yep, we can see it.
Okay, great. So we set up a whole bunch of these small tubes. And in each of these tubes is the DNA of the fish that we're interested in looking at, as well as several different enzymes and reagents. And so what we do is we set up a whole bunch of these tubes, like 20, 30, 50, depending on how many we want to look at. And then we take these tubes and put them into one of these machines I've got here on my right. So these machines are called polymerase chain reaction machines, or PCR machines for short. And all we do is we put all the samples we're going to look at on this block, and then pull the lid across, and then we just press go, like on a DVR machine. We just have a little green arrow that we press go.
Cool.
And then within 2 to 2.5 hours, we get many, many millions of copies of that individual gene that we're interested in looking at between the migrants and the residents to see if we can find any differences.
Wow, millions of copies. So that is one serious copying machine you have over there.
Right.
So what have you learned about the fish so far?
Sure. So one of the most interesting facts we've found out is it doesn't seem that there's only 1 or 2 genes that are different between the guys that stay at home and the guys that migrate. It seems that there are many hundreds of genes that seem to be associated with this behavior.
And so how will what you are learning impact the future to come?
Sure, so there's 2 real points. Firstly, on a real broad perspective, we still don't know how complicated behaviors and complicated traits are inherited. And the example I always like to give of that is human height. So we know that tall people generally have tall kids, right? Okay, but we still don't know how many genes or which genes are actually determining height in human beings. And it's the same kind of thing with migration in salmon. There are lots of genes, but we still don't know exactly which genes and what the identity of those genes are. The second thing that hopefully this research will be able to help with is a conservation angle. So unfortunately in the lower 48, so in Washington, Oregon and California, a lot of the populations of the migrant fish aren't doing really well. Their numbers keep going down, 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 down. And it's possible that some of the work we're doing here at Purdue is going to help us understand why that may be.
Gotcha. Well, Matt, thank you so much for joining us from your lab here at Purdue. You really work in some interesting places, doing some interesting stuff. Before you go, I hear we've got a hot seat question for you, so let's check in with Abby.
Great.
Abby.
Matt, the hot seat question of the moment is, do you know which type of fish live the longest, stay at home, and the ones that travel?
So in the species I work on, the migrants tend to live longer than the ones that stay at home. So migrant fish, when they leave the freshwater, they tend to be about 2 years of age. And then they migrate out into the North Pacific and spend about 3 years or so basically just swimming around the North Pacific and eating before coming back to spawn. So typically the migrant fish live to be about 5 or 6. It depends a little bit on the population, whereas the resident fish don't tend to live as long. They tend to live about 3 to 4 years.
Gotcha. Okay, and we just had an email question come in for you, Matt. This is from River Valley Middle School in Indiana. Are all diseases somehow caused by our genes?
Ooh, so no, certainly there are a lot of genetic diseases that are caused by mutations in individual genes, but there are a whole bunch of other diseases that are caused by pathogens, so things like viruses and bacteria and fungi that are out in the environment. So when you catch a common cold, for example, right? So that's from a virus, that's from a foreign body that's found in the environment that you ingest, and that manifests itself as a common cold. But there are a whole bunch of genetic diseases that are due to mutations in genes.
So it's sort of a
so
mix.
It's both, yeah, right.
Okay, let's go ahead and check in with our school Hales Corners. Do you guys have any questions for Matt?
Yeah. Certain types of fish easier to study than others?
So, yeah, sure. Some types of fish are really difficult to study. So there is loads of species of fish that are found really in deep waters in the oceans. And trying to get samples of those fish is really, really difficult, if not impossible.
Yeah.
One of the nice things about working on salmon and trout is when they come back to spawn, a lot of the times these rivers, as you saw in some of the videos from Alaska, they're not very deep. So it's really easy for scientists like myself to catch a large proportion of those fish that are coming back to spawn.
Right. Okay, and let's go ahead and check in with our studio audience. Do you guys have any questions for Matt? Yes, you sitting on the floor.
Have you found any dominant genes that, like, when the migrating fish and the resident fish mate, have you found like a dominant gene that will find if the offspring is going to be a resident or a traveler fish?
Did you hear that?
Yeah, I did. So that's an excellent question. So some of the work that we're doing here in Alaska seems to be that there is some dominant genes that seem to be associated. And the reason we think that is when we do crosses, so you saw some footage there of us doing crosses, so when we get the eggs from one fish and the sperm from another fish. If both of your parents are migrants, you are more likely to grow up to be a migrant fish than you are to be a resident fish. But that association isn't 100%.
Gotcha.
Okay, so even though you've got two migrant fish, a male and a female, when they breed, they still can produce and do produce some resident fish. So we think it's a mixture of both dominance and also co-dominance that's kind of explaining this variation in behavior.
Very interesting. Great question.
Yeah.
All right, let's check back in with Abby in our hot seat to see what question you have for Matt. Abby.
Right.
Matt, the question is, if you can breed fish and one would be a freshwater fish and the other parent would be a saltwater fish, would its offspring be able to live in both environments?
Ooh, hybrid.
That's a good question too, and that kind of gets at the question just now. So when the migrants come back, we can do that. We can spawn resident fish and migrant fish and see what the proportion of the offspring are going to turn into. So will they go back out to the ocean or will they stay in the fresh water? And like with the previous question, it seems to be a mix. So you seem to get some individuals that will stay in the fresh water, whereas others will migrate. So it's definitely a mix of the different sort of genetic traits. There's also in the environment is important. So if you warm the fish, if you turn the temperature up on the fish as their eggs, you get a higher proportion staying resident than you do migrating out to the ocean.
Gotcha. So it's sort of like humans. I mean, some people are more like their moms, some more like their dads, some like both. We just had an email question come in for you, and this one is from Newbridge Middle School. Or I'm sorry, St. Patrick's School in Indiana. Does the sex of the fish determine whether they migrate?
Oh, that's a good question. So potentially. In our population in Alaska, what we're seeing is that more females make the choice to migrate versus the males that decide to stay at home. Now that is really, really population specific.
Okay.
If you look at other populations, it can be a 50-50, male to female. If you look at different populations, the higher proportion seems to be male. But in our group population in Alaska, it seems that mostly the girls are deciding to migrate versus the boys that stay at home.
Gotcha. And let's go to our studio audience again. Any more questions for Matt? Yes, you.
Like my friend said, with the dominance, say you get one that migrates farther away from the other one, they have two different migrating paths. If you breed them, is it more dominance to go one way as a parent or the other, or do they make a new whole entire migration path?
Okay, so if I understand it right, the question was if you have one that migrates further and you cross that with one that doesn't go so far, what do the offspring do?
Yeah, what type of migration path do they have?
Okay, that's a great question. So unfortunately, we still don't know exactly where these migrants go once they leave the stream that we work on. We know that they go around the North Pacific, so up to the Aleutian Islands, then across east to Russia and Japan, but we don't know how far one individual fish goes, and we don't know how far your parents go, if that will determine how far you go, if that makes sense. So, for example, imagine that your mom went to Japan and back. That doesn't necessarily mean that her kids are also going to go to Japan and back. They go somewhere in the North Pacific, but exactly where, we're still not sure. That's a really interesting question. We'd love to find that out.
Okay. Wow, we've had so many questions come in for you, and we're going to have more at the end of the show. So thanks, Matt, for your segment, and we'll see you in just a little bit.
Sounds great. Thanks, Jessica.
Thanks.
Science is fun. It helps you to learn. You discover the marvelous interrelationships between all living things. You find out what makes things tick. Everything from a molecule to a living organism. In the study of science is found the most useful and satisfying knowledge of man. Why study science? Study science because you and Betty and the Nancys and Bills and Joes and Janes all over the country will find in the study of science a richer, more rewarding life.
Wow, that was pretty cool. An animal's genes and heredity are determined by its parents. Now that goes for dogs, humans, fish, and even salamanders. Get ready because we are going to solve a salamander puzzle. With us here today are Purdue wildlife geneticist Andrew DeWoody and Rod Williams. Hey guys.
Hi Jessica. Hey.
Now Andrew, can you tell us what it means to be a wildlife geneticist and what kind of work you do?
Sure, yeah. Wildlife geneticists are biologists who generally go out in the field, collect some sort of tissue sample from an animal. Maybe it's a fin clip like you just heard from Matt, or maybe it's a hair sample from a bear. Bring those back in the laboratory, get DNA from them, and use that DNA to study different aspects of their biology. We do a lot of work on eagles, as you can see here.
Oh, so cool.
Yeah, so we collect naturally shed feathers from the eagles, get DNA from those, and then from those feathers alone, we can determine the sex of the donor eagle. We can attach a DNA fingerprint that's unique to that individual bird, and then we can monitor populations over time and figure out who mates with whom, and if the population's growing or shrinking or so forth, just from studying the feathers and the DNA in those. And so it's wildlife geneticists use DNA to study secretive animals.
Awesome. Secretive animals. So you guys also study another mysterious or secretive creature, and that is the salamander, right?
That's right.
Rod, can you tell us about salamanders?
Sure.
Where do they live?
First, there's a lot of different species of salamanders, over 600 worldwide.
Wow.
And while they can be very similar, they also have some very striking differences.
Okay.
And I actually brought 3 species with you with me today to show you some of these differences.
Great.
So this first animal is a museum specimen, so it's not alive.
Yeah.
This is a small salamander, one of the small ones.
Tiny.
This is an eastern redback salamander. It's a woodland salamander, occurs in forests and woodlands throughout the eastern United States. It's a fully terrestrial salamander.
Cool.
So it spends its entire life on land.
And that's fully grown?
That's fully grown.
Okay.
Other species can be quite different.
Wowzers.
This is North America's largest salamander. This is an eastern hellbender.
Whoa.
This particular species is fully aquatic. It spends its entire life from a juvenile all the way up to an adult in the water.
Awesome. And we see some of your work there. And then who else do you have with you?
This last species is our eastern tiger salamander.
It's alive!
This is a species that spends part of its life in the water and as a juvenile and part of its adult life on land. They only go back to water in the spring to try to find a mate, lay their eggs in the water.
Very cool. So if they're so secretive, how do you guys actually find them?
Well, it depends on which species you're talking about. If you're talking about the terrestrial woodland salamanders, you go into the woods, you sort through the leaf litter, you roll over logs. They're very secretive and cryptic and hide under structures. Hellbenders, as you saw in the video, you have to put a mask and snorkel on and swim underwater and try to find them under rocks.
Oh, wow.
This particular species, you can intercept them on the way to their migrations, again, to find a mate and lay their eggs in the water during the breeding season.
Awesome. So you guys know a lot about them, so I have to ask, what are you trying to find out about tiger salamanders exactly?
Well, we do know a lot about them, but there's still many things we don't know about salamander biology, in part because, as Rod mentioned, many species lay their eggs in ponds that are often really murky. All of the breeding biology occurs in situations that are very hard to observe. So what we can do is use genetics to figure out questions like who's coming to the breeding ponds, who's breeding with whom, and determine things like parentage.
Parentage. So can you define parentage for us?
Yeah, parentage, sort of intuitively, we all know that that everybody in this room and out there watching has a mother and a father.
Right.
And it's the same way for virtually all wild animals. And so what we don't know is the pedigree of these animals. Who was this salamander's mother? Who was its father? And how do they choose their mates in the wild? And so we can use genetics to try and sort that out.
So why does determining parentage matter?
Well, it matters in part because mating systems and parentage determines the genetic variation in populations. And genetic variation is important because it helps determine how populations can respond to changing environments. Everything from pathogens like Matt mentioned earlier to things like climate change. And so the genetic variation of the population allows it to evolve in response to a changing environment. Parentage can help determine that.
Gotcha. This little guy was trying to steal the show for a second there. So how do you guys actually collect your data?
I think we've got a video of Andrew and myself, along with some of our graduate students out in the field, trying to collect some of the data that we're going to talk about here a little bit later in the show.
Awesome. Let's take a look.
The Purdue Wildlife Area is used for a large number of different research projects. One project in particular is a project examining how tiger salamanders, which are amphibians, breed. So what you're looking at here is a drift fence. This is a technique by which we can intercept amphibians on their migration towards a breeding pond. And so many amphibians will overwinter in grasslands or in forests, and every spring they'll leave their overwintering sites and migrate to a pond, what you see behind me. We'll dig a trench, we'll take the fence, we'll bury the fence into the soil, cover the soil back up. Salamanders and other amphibians will walk down the fence, not see the pitfall traps, fall into the traps, and they're captured and held there until researchers can come along and pick them up.
Look out!
Researchers will come every morning and every afternoon and check the traps to ensure that the animals aren't held in the traps longer than necessary. So there are several ways of getting DNA from a salamander, and one of the ways that you can get DNA is to take the tail and take a small tail snip from the tail, and the tail will regrow or regenerate, and it's relatively painless to the salamander.
Well, this is a tiger salamander egg mass. If you look closely, you can actually see the individual embryos within this egg mass. We can take a small tissue sample from these embryos, in addition to the tail snips from the adults, back to the lab, extract DNA, and then conduct the parentage analysis.
Cool. Well, now that we have the DNA from the salamander's tails and their eggs, we need your help in figuring out the parentage of these salamanders. So, Andrew, you're going to take us through a Punnett square activity to help us determine who the parents are.
You bet.
Okay.
Yeah, we've got some video to help us with that. So
Awesome.
Here we've got a cartoon that illustrates, we captured all of these adult salamanders that have migrated to the breeding pond. We caught them in the pitfall trap, took the tail snip. We've also got all of the egg masses and the embryos in those, and we extract DNA from all of these individuals, and then we assign a genotype to them. And as you can see here, we've got 6 different embryos that have been genotyped. Half of them are CCs, half are CDs.
Okay.
And then we can compare that to all of the adults, and you can see their genotypes have now been determined and added in there. And then we can go through all the possible combination of parentage, so every possible male with every possible female. And so here's the first example. We have a dad who was AA and mom who was AA, and as we learned earlier with the Punnett squares, that gives rise to embryos that are all AAs, and that's not what we saw in this egg mass.
So that's not a match.
That is not a match. Okay. That is not the mom and the dad. Well, we can then do that for every successive parent pair until ultimately we find out who were the parents. And in this case, we see here we've got a dad who was CC, a mom who was CD, and our Punnett square shows that half of the offspring should be CCs, half should be CDs, and that's exactly what we saw in our egg mass. So those are consistent with parentage of the embryos in that particular egg mass.
Yay, we have a match. Okay, so let's talk about those results then. What did you guys find out in your salamander parentage study?
So this is a great example to show how wildlife geneticists can use genetics to unlock mysteries of secretive species like salamanders.
Right.
And we've actually learned quite a few things new about them using genetics.
Okay.
First is we've learned that females will actually lay their eggs in many different locations in those wetlands that you saw in the video.
Okay.
And within those egg masses that are distributed all throughout the wetland, they may have different dads within that egg mass.
Gotcha.
And more importantly, those different dads are chosen by the female based on their size.
Really?
Females tend to prefer males that have longer, taller tails.
So how does this guy fare?
He's going to do very well.
Oh, he's such a go-getter. I'm so proud of you. All right, so I have to ask you guys, how did you guys decide to study salamanders? Don't you find them a bit creepy to work with?
Oh, not at all. I don't think they're creepy, and they're a great example of a secretive species where it's hard to know much about their biology because of their way of life, and DNA offered a great opportunity to learn more about their biology.
Very cool. Okay, well, out of our studio audience, I want to know who does not find a salamander creepy? Anybody? Anybody want to come touch it? You. Come on up here. And what's your name?
Austin.
This is Austin. So I want you
Austin.
to touch the tiger salamander. How does it feel?
Slippery and wet.
Slippery and wet. Do you like the look of it?
Yeah.
I think it's pretty cute. Oh, so cute. Okay, thanks, Austin. Now let's go ahead and check in with Abby to see what's happening in the hot seat. Abby.
Hot seat users would like to know, how do you extract DNA out of hair or feathers?
Well, it's much like Matt talked about the process of PCR involving different enzymes. And so we can take that hair or sample or that feather sample or a fin snip back into the laboratory. We can incubate it at fairly high temperatures and use some enzymes to disrupt the cell membranes and release all the DNA that's in the nucleus. And then basically we chop everything else up, all of the carbohydrates and lipids and everything else, until we're just left with the DNA.
Gotcha. Okay, and we just had an email question come in for you from St. Francis and Clare School in Indiana. If the tiger salamander was migrating, where would you catch it?
They actually have a very wide range of habitats in which they live. They can migrate from a forested setting, a grassland, and you just put up a fence like we showed in the video and intercept them on warm, rainy nights in the spring, which hopefully we'll have here in the next couple of weeks in Indiana. And they'll migrate in mass numbers. They're aggregate breeders, means hundreds of those guys will migrate to the pond to breed.
Awesome. And now let's check in with Hale's Corners. Do you guys have any questions for our scientists?
Yeah. Can some salamanders be poisonous, and will it affect the research?
Can some salamanders be poisonous, and will it affect their research?
Many species of salamanders will secrete a slight toxin from their body, and they use that to deter predators. So yes, they're in the level of toxicity of those secretions varies from species to species.
Cool. All right. And let's go ahead and check in with our studio audience. Do you guys have any questions for Andrew and Rod? Yes, you all the way back there.
How long would it take to find out the DNA inside like a feather? Like how long would it take?
How long would it take to find out about the DNA from a feather of one of the eagles?
Yeah, not too long. Maybe it takes really just a few hours to get the DNA from the feather, and then the actual genotyping part, that's necessary for our Punnett square maybe takes, a few more hours or another day. So within 2 days, you can learn just about everything there is to know genetically about a feather.
Awesome. Yeah. Well, you guys do great work, and we're glad to have you, and we'll see you at the end of the show. And remember, if you think of more questions out there, you can submit them through the It's It's a Gene Thing page on the website.
Hi everyone, welcome back to the show. Our next guests have a big question to answer. Say hello to male and female tiger salamanders, Sal and Amanda.
Thanks. Nice to be here.
So, what brings you here today?
We're here to learn if Sal is the father of my embryos. I'm not.
Here's a shot of the egg mass in question.
See? They look just like him. Just like him. No, they don't. No, they don't. You don't know me. You don't know me.
Well, we've run the genetic test to determine the parentage of these embryos. Let's see the results. Sal, the genotype doesn't match. You are not the father.
Oh, that cracks me up. Now, we hope that you've had a great time meeting some real-life geneticists here at Purdue University and have learned some things too. Remember how animals can have different characteristics because of mutations, like the mice with different coat colors. Also, geneticists travel all over the world to learn more about what causes animals like fish to move from place to place. And you can use scientific inquiry to help solve important heredity questions about animals like salamanders out in the wild. Now, we do have time for some more of your questions for all of the scientists in today's show. So first, let's go to the hot seat. Abby.
Hot seat users have a question for Andrew and Rod, and they would like to know if you breed a venomous salamander and a non-venomous salamander, what would happen?
Well, to my knowledge, there are no venomous salamanders.
Okay.
And the toxins that we were talking about earlier are species specific, so you can't really interbreed those different species.
Gotcha. All right, and we just had an email question come in for you, Nadia. This is from New Bridge Middle School in North Carolina. Hey, North Kakalaki. Why do plants have different colors, even different colors on the same plant?
Well, there's a lot of different ways that can happen. So first of all, plants make a lot of secondary metabolites and a lot of different compounds in plants. So purple a lot of times comes from a chemical called anthocyanin. And so, so you have a lot of compounds like that, and then sometimes the different patterns you see on plants can even be caused by what people refer to as kind of jumping genes. So they're called transposable elements, and they can move from one place to another in the genome and actually cause the different colors in the kernels you see in corn and even different patterns on leaves. So there's a lot of different ways it can happen.
That sounds really interesting. And now let's check in with Hale's Corners. Do you guys have any questions for any of our scientists?
What made you want to get into genetics?
Who wants to take that? What made you want to get into genetics? Anybody?
I mean, I'm happy to say that. I think it's just a great technique for somebody like me who likes to study wildlife. I mean, it was sort of the latest, greatest technology. You know, you could go around and collect these samples, bring it back to the laboratory, and answer questions that we really had no idea about until the advent of genetics. So just like salamander parentage, nobody really knew that females partition their clutches. They didn't know that they mated with multiple males, or that the females were really interested in tail height and tail length to the extent that they are.
Right, and now we do, and you'll continue to learn more and more about things you don't know about right now.
We hope so.
And what about our studio audience? Do you guys have any questions for our scientists? How about you?
Mine's for Nadia, and with the square, I forget the name of it, how big normally does that get for just maybe one species of plant, human, or animal?
For the Punnett square? Okay, so it depends on the question you're asking. So some traits that you're looking at, maybe you'll have only like big A, little a traits, or big A, little a, big A, big A. So, but you can also have traits that have like multiple genes that kind of go into the phenotype, and then your square gets bigger. So it depends on what question you're asking, really.
Cool. All right, and then let's go back to our hot seat, Abby.
Hot seat users have a question for Matt. Do you study any animal other than fish?
Right now I'm only studying fish. In the past I've studied different species of fish as well as birds, insects and even pine trees. So I've studied a whole bunch of different organisms, but currently right now my research is really fish oriented.
And is that your favorite?
Yeah.
Yeah. All right, and let's go check in with Hale's Corners. Do you guys have any questions for our scientists?
How many places have you gone to work? How many places have you gone to study fish?
Okay, that's for Matt. How many places have you gone or traveled to study fish?
Okay, so there's the work I've done in Alaska, as well as work we're doing in the Great Lakes with the first species you saw in the first video, the brook trout. I've also gone up to Lake Oneida in New York with Andrew actually, working on lake sturgeon, which is a project I did here at Purdue before I started to work with salmon and trout. So those are the three main places I've been to work on fish.
Awesome. Well, for the questions we were not able to get to today, we will be sending out an email with the most frequently asked questions from today's show for teachers to review with their students. And for future zipTrips, keep checking those emails and the zipTrips website for updates. Thanks to our students here in the studio and our scientists. For all of our zipTrip crew, I'm Jessica Jackson. See you next time. Bye.